Thank you. Thanks for the invite today. Yeah, time for something a little bit different, I guess. So today I'm going to talk about really new and exciting technology which could potentially be applied to RNA platform and lots of other different types of preclinical research. And so off we go. If I can get the clicker to work. Yeah. So really the organ chip work has really been driven by some really rapid improvements and leaps in bioengineering, which will essentially allowed the miniaturization of the traditional cell culture into some very small chips you see there, about the size of a USB drive. And so in terms of ESR, we think that these new chips will have a really wide range of use across Aotearoa and lots of different applications, such as RNA platform, and with lots of different impacts in different areas. And so what we do know in terms of drug delivery, that animals have been used for more than 80 years to test the safety of medicines, but what we also know that they're actually not a very perfect model, and as it says that they're almost a third of the medicines that test and look really great in animals actually don't translate to being useful in humans, or indeed may not even be safe. And the other aspect, I guess, in terms of animal testing is now there's a heavy move towards the 3 Rs in terms of reducing and replacing and refining the use of animals. Obviously, there's a wide sentiment in terms of the public now about not wanting to use animals in research. Okay. So what is an organ chip? I'm sure many people here will be familiar with the old culture plates that have been used for many, many years, and these have served us pretty well, but as you probably will see, if you grow something in a petri dish, it kind of looks like a carpet, and last time I looked, people's organs were not flat. So what we really want to do is try and mimic the structure of cells, because as you can see up there, particularly some types of cells like epithelial cells, endothelial cells, they have a very defined structure which contributes significantly to their function. And so many of you will be aware of some of the new and advanced sort of approaches. Organoids are certainly becoming very, very popular. Spheroids is another 3D system that's being used as well. And there's also been a bit of work now which is kind of trying to elevate the transwell system by sticking 3D structures in that. But what we're really at ESR most excited about is the flexible organ-on-the-chips which use microfluidics. Okay, and so what do the chips actually look like? So there are several different types of chips which I'll briefly touch upon, but this is the main type of chip. And so we have basically 2 channels, a top channel and a bottom channel, and in these channels we can flow media through them at different rates. Between the 2 channels, they're actually joined together through a semi-permeable membrane, and one of the really cool things is that these chips being flexible means we can apply different types of stretch on them, so we can actually mimic sort of the intestinal movements or breathing of the lung, and again that adds in terms of gene expression and things like that, much more closely mimics things than the old traditional systems. And if you can just have a look on the right hand side up there, this is kind of what the standard system looks like. As I say, we have a sort of larger top channel where we can stick the epithelial cells and flow the media or reagents across there, and on the bottom side we have the endothelial cells we can seed in there, and they kind of grow all around the channel, and that I guess kind of mimics the blood flow to the system. And so when we seed the cells into these chips, as you can see from the image there, what we actually get is a form of the cells and physiological structures which much more closely mimic those of the human body rather than the cells. So we get things growing up in these completely random and really cool and interesting projections growing everywhere. Okay, and so this slide here, I guess, is just to illustrate that there are many, many different types of organ chips in development at the moment, and there are also moves to be able to join some of them together as well, which is obviously going to be important, but at the moment, most of the organ chips are just a single organ at a time. Okay, so what does this mean in terms of how we can use the organ chips? So there are several different areas. So we can look at drug development and vaccine, preclinical testing. There's also obvious uses in terms of precision health. If you can take people's primary cells, you can develop personalized medicines for them. Something that ESR is obviously very interested in is around toxicology and environmental applications. We can better understand disease mechanisms, and obviously things like gene therapy and immunology can be undertaken quite easily in this highly controlled system. So just in terms of what's the journey that we've been on in ESR in terms of developing this organ chip technology. So back in 2019, we sort of saw the emergence of these new 3D systems, and we thought it would be a great idea to bring them into ESR. So in early 2021, we got the Emulate system, which is still arguably the leading system for organ chips in the world. And so we worked on that for a couple of years, and then in 2023 we kind of decided, initially we were mostly focused on the intestinal model, but now at 2023 we decided to widen the program and start looking at different models, and also now we're trying to open up the platform so that other people can maybe engage with us. So just to give you an idea of the applications that we're doing at ESR at the moment. So we have three main divisions at ESR. So forensics group are really interested in looking at toxicology and pharmacological applications and looking at the mechanisms of drugs, particularly new drugs which may have harm. In the environment group, there's applications around food safety, risk assessment, looking about how pathogens can interact with different organs. Also chemical hazards, which is a little bit of an understudied area. The health group, obviously really interested in seeing if we can develop specific models for Māori to see how they may be impacted by various hazards. And they're also interested in the precision health type things. I should say there, we are opening up the platform now. We are looking for collaboration opportunities. I must say here, I do have to take a plane really quickly after this session, but you can hunt us down or ask the RNA platform for our contact details. Okay, so what does it look like? So the main models we're looking at that we have developed at the moment primarily is the gut chip, as I mentioned before. This is using intestinal cells. And why is this important to New Zealand? Well, New Zealand has a really high rate of foodborne disease compared to other developed nations. So we have been testing foodborne and waterborne pathogens, looking at things like pathogenicity, inflammation, human responses to infection. And just that slide up there is kind of illustrating some of the work that we've been doing around Campylobacter. So many people would have known about the really bad Havelock North outbreak that we had, of waterborne Campylobacter outbreak. But through this organ chip work, we've actually been able to look at the different isolates that were from that outbreak, and we've been able to look into the organ chip and test which ones can invade into the cells. So often the genetics tell us one thing, that they have the genes for invasion, but actually proving that they do invade cells is a key step to understanding the disease pathogenesis. Another area that we've been developing is around the lung chip, and as I said before, There are several different types of chips being developed, and so this is a really new chip which allows us to look at aerosols. So we have a PhD student who's really interested in looking at vaping. Again, a big issue for New Zealand in terms of youth. So looking at things like e-liquids, synthetic cannabinoids, and toxicity, DNA damage, and inflammation. The third model that we have up and running is liver chip. Again, in terms of ESR scope of operations, we're really interested in synthetic drugs, which, as it says there, kills at least a dozen people per year. So looking at how drugs may harm the liver. Also, there is some work to do with pharmaceutical development, and obviously this is an area that also interfaces a bit with the RNA platform. So yeah, in terms of the models and value to the RNA platform, we can provide proof of principle, mechanistic and efficacy applications. So you can do the ADME studies and preclinical safety and toxicological testing as well. And just finally, there's a couple of slides which are probably quite applicable to the RNA platform. And so Moderna has been looking, as many of you will know, at developing lipid nanoparticles. And so they did a study on rodents and find everything was fine with their nanoparticles, but when they moved it to primates, they found that there was severe liver damage. So then they've gone back and worked with Emulate, and they found that the liver chip predicted the damage in the primates really well, and not only that, it was cheaper and quicker. And finally, I just mentioned there's just been a very recent study on another model, which is the lymphoid model, and so again, this is a collab between Emulate and Moderna, and what they've essentially shown is that they can put cells in myeloid cells in the chips, and they can flow the spike proteins from COVID over the top, and they can get the correct responses being made. And I guess most importantly, this also works with the lipid nanoparticles as well, so they can deliver the RNA to the cells within the chip and get a correct physiological response. And I believe that worked with both the Moderna and the Pfizer lipid LNPs. Finally, yeah, I should acknowledge people, particularly Jan Powell, who works closely with me to develop this platform, and a whole lot of other collaborators. Thanks very much.